There's a need to start up. Okay, I think it's really great to see that you have such a great time. It's good to be together again and meet at the scientific meeting. So, but now we have a new speaker with a bit of a tight schedule. So I think there is a need to introduce him. We will now be we are extremely lucky to hear from Marcus Claesson. He's from Sweden, but he is currently in Cork and unfortunately was not able to travel here today. But it's really perfect to see that you are able to join us online. He has a master in bioinformatics from Gothenburg, and later on he moved to Cork and has a PhD where he has been working on, seems like probiotics and so on. And he is now a lecturer at the University of University of Cork. And I think if you go in the literature, he is the first author of one of the, I think at least an eye-opener and a landmark paper on the gut microbiota in elderly. It was published in Nature some years ago, and how it's influenced by diet, but it's also associated with health outcomes. So now now we'll hear about his current research, which is a lot about IBD and the microbiome, and we're happy to hear if it can predict its course. So go ahead, Marcus. We're looking forward to it. Thank you.
Okay. Thanks for that kind introduction. I'm delighted to be able to present to you today, and I'm sorry I couldn't be here in person. And thanks to Hanne and Johannes for the very nice invitation. So, as was just mentioned there, I'm originally from Sweden in Gothenburg, but I've been nearly 20 years at the south end of of Ireland in West Cork, and I've been doing my PhD, postdoc, a bit of other work experience at the University College Cork and APC Microbiome Ireland. And I did indeed work on the microbiota in the elderly, and some time ago, but then as PI in my new group, we focus more on inflammatory bowel disease and the role of the microbiome. So that's what I will talk about mainly today, to quite recent papers that we published on this, to different studies on IBD, but from different angles and with different results. So just a quick disclosure. So I'm the part-time CEO of a company, Seek Biome, and was a consultant for Second Genome and Mars Pet Care. Okay, so a brief introduction to inflammatory bowel disease. In case you haven't heard too much about it, it's made up by Crohn's disease and ulcerative colitis, have 7 million sufferers worldwide, and only in America is the personal and healthcare system costs are about 32 billion. They're improving medication for these diseases, but there's currently no cure unless you count surgery as a cure. They manifest in a little bit different ways. So you have here as a it can be either proctitis, left-sided colitis, extensive colitis, where the red part is the inflammation. It doesn't go that deep into the tissue, whereas it does for Crohn's disease, which also can go further up in the intestinal tract. You have here in the ileum as well, and it's more patchy in its nature. And it's a multifactorial disease, so it's genetic factors, immune response, and lots of environmental triggers, including diet and lifestyle, can give rise to IBD as well. And my particular interest is the change of the microbiome, which I'll talk about next. So the first study we published last year was based on the biopsies from the colonic microbiota. And we took from 161 subjects, so UC, CD, and healthy controls. We took 2 biopsies at the same time point, one from the inflamed region, one from the non-inflamed. And because we collected the samples in RNA later, it was possible to not just look into the microbiota, but also the host transcriptome, epigenome, genomics, and genotype. And we also had diet food frequency questionnaires on this. So this is a PCA, which is quite commonly used in the field of microbiota. You have every person is a line, and if the ring, the hollow ring here is the person's inflamed biopsy, and the triangle is the non-inflamed. So you can see the microbiota isn't too different from lots of them here. So they're quite subject not too many even subject differences here. And the ellipses are confidence regions. 80% of the samples are within these ones. So you have red controls, yellow UC, and blue Crohn's disease, where the dotted line is from the inflamed samples. And these two principal component axes correlate well with, so you have higher alpha diversity over here if you call it Plasmodium. If I call it bacterium Plasmodium, you also have E. coli, Ruminococcus, and a few others were known to be associated with IBD over in this end. What we also see is that you have this kind of gradient of inflammation. It's not, they're quite overlapping these regions, but you still have slight shift away from healthy controls to non-inflamed UC, to inflamed UC, to non-inflamed CD, to inflamed Crohn's disease in this way. And if you look into the alpha diversity, so within each sample, you can see here, which follows the literature, that it's significantly higher in healthy controls compared to Crohn's and UC. I mentioned this gradient inflammation, which is kind of overall globally there, even if it's quite moderate. But even when we do subtractive changes along PC1 axis, we can find that when you subtract inflamed values on the PC1 axis with a non-inflamed for each person, that is significantly above 0 for Crohn's and UC. So it's within patient as well. Now, the next obvious thing to do was to look into particular taxa and whether they were significantly higher or lower abundance for these various diseases. So this is our volcano plots. So you have the significance on the y-axis and the log2 fold change on the x-axis, and any taxa that goes above. So these dots are individual like species generally. They are, if they go above this line here, they are significant. So we didn't find any associated taxa associated with inflammation, even though we had a slight shift from the previous slide. We did, however, find particular taxa, unclassified Lectinosporisa and Anorostipus hadrus that were higher in healthy controls compared to Crohn's disease or ulcerative colitis. So we're currently looking into in vitro and in vivo experiments for these particular strains that we want to identify and find out more about as potential beneficial microbes for IBD. Okay, so this very colorful plot here shows a heat map on microbiota only. So each line here is a sample, and they're divided by the CD healthy and UC here according to these colors, just on the microbiota, and we divide them into 10 subclusters. And you can see each of the 10 subclusters have representatives from each of these cohorts, even though there seems to be more red healthy over here, which also coincides with more, these are taxonomic family classifications down here. You can see a lot of variation here as well, but seems to be more red Firmicutes families over in this end. Some clusters are nearly completely dominated by Escherichia shigella in green here. And we also have Prevotella copri over in the more healthy parts here. So lots of heterogeneity. So it's not very clear cut in this way. And you can see, if you look at particular clusters, you can see that they're dominated by particular taxa. Bacteroides fragilis here, Romanococcus gnavis, vulgatus, and Dorii in here. And we could find that some of these representative strains from some of these were pro-inflammatory in vitro. I mentioned earlier that we collected the biopsies in RNA later, so we could look into the epigenome, the host epigenome and the host transcriptome, and we compared subjects from each of these clusters to see if that was different for the epigenome and the transcriptome, if that was different from others. So we have represented some genes here, like NOTCH4. It's hypermethylated, so it's likely to be downregulated based on the epigenome in cluster 1 compared to all the other clusters. And you have DRAM1, same thing there, that's hypermethylated. You have, and also in some cases we have where you have the methylation, DNA methylation, and gene expression coincide. So this TRIM27, which is known to be involved in promote inflammation in colitis. That's upregulated down here in cluster 2, and it's hypomethylated in the same cluster, the same gene here. So there's some interesting things here to follow up. We also looked at a more global view of the epigenome. So this is a PCA of the host epigenome only, so not microbiota. And you can see here that we only have the epigenome for Crohn's disease and healthy controls, by the way. We didn't have it for ulcerative colitis. And you can see here that if you look into the principal component 1 axis, you have a clear shift from the non-inflamed Crohn's to the inflamed Crohn's here, similar to what we saw with the microbiota. And if you go down to look at the 6th principal component, we also have a shift between Crohn's disease and healthy controls as well. So there's clearly a stronger trend with inflammation with epigenome and a slightly weaker, but still there, trend in terms of disease. Okay, so because we had so many different types of omics data, we want to be able to see if we could classify disease states and inflammation status based on combinations of these omics data. So my previous PhD student Anna here, she used XGBoost, which is a quite powerful machine learning algorithm on various combinations. And you can see here in red, so that the end combination of epigenome and microbiota were the best classifying CD from UC and CD from healthy as well. And same thing for inflammation status. So any red line here, the area under the curves, if that's the closer it is to 0, the better it is at classifying. Anything that's with the epigenome is added to seem to improve this classification. So this is part of the study, and currently we're following up based on the same biopsies and the same similar data, because these biopsies were taken nearly 10 years ago now, and we have looked into patient letters and between the general practitioner and the clinician to see if these patients went into relapse several years after they gave the biopsy. So this is a kind of very interesting way of seeing how these data potentially can predict relapse. So we have this data for 116 IBD patients. And Jill here and my collaborator Andrei Temko, they looked into combinations of omics data. We have 6S DNA, so this is the standard gene, and we also have our RNA, so that's a transcript, so which is possible to have because we have RNA later. And then we have host genotype and age as well. So what Jill found was that there's quite good predictive power here in area under the curve of 0.75, around close to 0.7 with some other combinations, but the best combination was if you combine genotype data with 16S rRNA and age. And then with 0.75 area under the curve, it's possible to predict if that patient within 2 to 4 years went into relapse, just based on a previous biopsy. So this is something we're following up and hope to publish soon. Okay, so that's the first study I want to talk about. The second one is also on IBD in the microbiome, but this time based on stool samples. So it's possible to collect many more samples due to this non-invasive sampling technology. We had samples from both Cork and Manitoba in Canada. So we had about 700 subjects at 3 different time points with 3 months apart. And this is a PCA with the same color coding as previously, but now we're looking into temporal dynamics as well, because many of these patients went into relapse or remission. They went back and forth a little bit over the course of the 6 months. So you can see here that healthy controls were more defined, less variable compared to ulcerative colitis and Crohn's disease. And also here you have a gradient of disease activity this time, away from healthy controls going in this direction, so even though it's quite moderate. As expected, we found that the alpha diversity was lower in Crohn's and UC than healthy controls, just as before, but there was no significant difference between disease states, disease activities in terms of alpha diversity. We did, however, find that people who, during the course of the study, transition between relapse and remission, so between active and inactive. So you have this transition subjects here. We looked into the microbiota, the beta diversity between those subjects or within subjects, and compared with those that only stayed in active or inactive, those were healthy controls. And we found that those transitioning had a higher microbiota diversity overall between them, between sampling points from the same person. So the microbiota was less stable, which is kind of intuitive, but it was still nice to find. Similarly to the previous study, we also looked into differential abundance, see what taxa were up or down in comparison to Crohn's disease and healthy. There's loads of taxa here that were, Eggerichella lenta is one of those that were up here, that were more common in Crohn's disease compared to healthy controls. And same thing for UC, although it's some differences between UC and CD, but there was no significant change between activity and between active and inactive Crohn's disease or ulcerative colitis. Or slightly bacterial story was slightly higher in active UC here. So again, there was a lot of overlap in the PCA on the previous slide. So this shift, even though there was a shift away between inflamed and non-inflamed, it didn't really translate to specific taxa that were higher or lower between these states. We collected a lot of metadata, part of this microbiome study, and we could rank these based on how much they explain the beta diversity difference in terms of Bray-Curtis. And we were not surprised to see Crohn's disease explaining most of this here, but we were a little bit surprised to see that geography location, whether the person was from Cork or Manitoba, played a big role, then followed by surgical resection, which obviously has a big physiological impact on the gut, followed by alcohol and ulcerative colitis and a few other diet and supplementary and drugs here. But even if you add them all up, they explain less than 10% of the total microbiota variance, and we think the rest would be stochastic or it would be individual variation. So, you know, all of us, our microbiota changes on a nearly daily basis, or it could be due to factors that we haven't measured. Here we also wanted to see if we could predict whether someone was in active or inactive stage just based on the microbiota. And you can see here that when we just looked at one time point, so OTUs, so the taxa abundances from one time point, this wasn't possible. So AUC was close to 0.5 here for both UC and CD. But, and this is where we got really excited, is that when we looked at 2 consecutive time points for each person, we actually did the ratio of OTUs, and we used these values instead. Then it was possible to get much better classification performance. So AUC was 0.73 and 0.81 for Crohn's here. So this is quite promising if you want to be able to predict relapse and maybe for diagnostic or prognostic uses. And this is something we're following up at the moment. I mentioned we had a lot of metadata on this, so we also looked into microbiota associations with various treatments. And you can see here that, so this is beta diversity plots here. It's the same dots plots as you saw on the previous slide, and these are alpha diversity into resection, if they took mercaptopurine, 5-ASA, or biologics, or proton pump inhibitors, or had steroids. So these are common drugs for IBD. And we found of all of these, the resection had the biggest impact on microbiota. It seems to be that Faecalibacterium prausnitzia and Clostridium leptum were down in resected patients, while Romainococcus novis and Bacillus fragilis were up. We also find that 5-ASA here, that people who were taking 5-ASA, that IBD, yes, they were closer to healthy controls than those who did not take it. And we think this the reason for this is that 5-ASA reduces the pH and might promote more health beneficial microbes, lactic acid bacteria, and so on. And also that people who take 5-ASA, they generally seems to be a quite mild disease, and that's one reason why they could have higher microbiota diversity as well. We also found that proton pump inhibitors, while not being a specific IBD drug that they had those who took that had lower alpha diversity and a slightly different beta diversity. And we're currently following this up in more granularity because we have dosage as well, and we have particular drugs. So these are just the broad drug categories, and we want to go into more details because we have such a huge sample size. Okay, so another study which is related to that was published a couple of years ago, but it was based on samples from the same cohort. We wanted, because we collected dietary information, we wanted to see if low fiber diets was associated with bacterial synthesis of butyrates, because Crohn's disease patients in particular are recommended to take quite have a low fiber diet. So we measured this butyrate synthesis by doing qPCRs of the butyryl coenzyme A transferase, or the B-coat as we call it, on a subset of these patients. And if we stratify the qPCR results of this gene based on stratified into those that have a high and low B-coat abundance, we can see that those that have CD active have fewer B-coat genes than CD inactive, and same or controls or ulcerative colitis. It also turns out that this stratifying group of B-coat less than 9.5 abundance, that they had lower microbiodiversity than those that have higher levels of this gene or controls CUC. And as we perhaps should expect, there's also higher levels of known butyrate producers like Rosburia, Butyrococcus, and FPRAU in samples that have a higher B-coat abundance. We also looked into the microbiota composition of these groups and found that the microbiota that was quite shifted for the low B-coat group here in red, and we did a PCA on the diet only. So this PCA is only to do with the diet, and that diet is quite shifted from the low B-coat group compared to all the other groups. And from kind of our hypothesis, why it turned out to be true, that this bacterial butyrate synthetic capacity seemed to be indicative of a high fiber diet, because patients who had a higher level of B-coat, they also ate more fruit, vegetable, wholemeal bread, cereal, and nuts than those that did not. So that's we think that's an important finding. And if you take a step back and look into the general association of diet and microbiota, one of the studies that was mentioned at the start was the one in elder methods in older people. And here we did we found strong association between microbiota and diet. So this is a PCA of diet alone. So you can see in green here are older people who live in a community, and their diet is very different from those who live in long-stay care here in red. Back then we also correlated the the alpha diversity on the y-axis with the healthy food diversity. So in other words, how healthy and how diverse their diet is. And we found a strict correlation here, and people who were living in community have higher of both. So we wanted to repeat this in the IBD cohort. So this is very significant, obviously. We wanted to repeat this in the IBD cohort where we took colonic biopsies. So that's the first study I talked about, and we found no such association. The diets completely overlapped, more or less. I think white bread was more common in IBD than healthy controls. That's the only difference. And the same alpha versus healthy food diversity was non-significant. But when we looked into the stool cohort, this paper published in Gut, based on many more samples, there was a slight shift of patients, of healthy controls in red here, in terms of the diet away from IBD here. And also, there's also some, overall, the alpha diversity had a slight significant correlation with healthy food diversity. So a little bit significant. So we think it's still very important to look into in microbiome studies to make records of long-term dietary habits, because it could affect the microbiota. It doesn't seem to affect the mucosal microbiota as much as the fecal microbiota, which in a way makes sense, because that's what is more, the stool microbiota have a higher response, more quicker response to what you eat than microbiota that are attached to the gut wall, really. So it's still very important to monitor diet in this way, I think. Okay, the last study I want to talk about, which is ongoing, so I don't have any results to show, we call it Predict UC. So we want to predict the course of ulcerative colitis or find biomarkers that predict relapse for UC. Here we have a much smaller group of patients, up to 30, but they are known to relapse frequently within 1 year, and we take monthly stool collections from them. This is all completed, but we're currently analyzing this. We look into the microbiome, the phagome, the mycobiome, so the fungi, and also metabolome. We measure fecal calprotectin and a few other disease questionnaires, and diet and perceived stress as well. So we hope to find a combinatorial machine learning model that can then be trained on these data so that it can predict whether someone is heading into relapse or not. And if that proves to be efficient, then those people can receive early medication. And this obviously needs to be validated in a bigger IBD cohort, which we already have in place, the one that I talked about earlier. So, okay, so now I want to change tack a little bit and talk a bit about EU cost action, which I'm chairing. It's called ML for Microbiome, and it's basically, I won't go too much into machine learning here now, but machine learning has huge implications for lots of science, including microbiome. The thing is that in order to predict and to be diagnostics and prognostics for health, machine learning really, rather than having very specific algorithmic instructions, like many other software solutions have, you train these algorithms with data where you know the answer, and then you test it with other subset of the data where you don't know the answer, and you improve the algorithm in that way. And that turns out to be quite a challenge for machine learning, for microbiome data, because there's so many different data types. You can have 16S, or you could have shotgun, for instance, and also subsets of these things. You can have various regions of the 16S genes, and this can lead to a lot of noise and variation between people. And another important challenge is that generally, if you want to use something like deep learning, which is a very powerful machine learning technology, then you need many more samples than when you have features. So features in microbiome data would be number of species, number of genes. So what we call a feature table generally looks like in microbiome you could have hundreds or thousands of rows. These are genes or taxa, or you have maybe 10, 100 or 200 samples, columns in this feature table, which are the various samples. It should really be the other way around to have efficient deep learning solutions to that. So that's one of the main challenges. So it's quite hard to generalize these machine learning models across studies. So the aim of this cost action is to first optimize and then standardize the best practice of using them for human microbiome research. So we're halfway through in this cost action. We have about 120 scientists, machine learning and microbiome scientists from 34 countries. We divided the cost action into working group 1, 2, and 3, and 4, where the first one is kind of a tech watch, where we keep an eye on what's in the literature and we evaluate those. And the second one is where we find benchmark data sets to test new methods on, for instance, and the third one then, which is a key one, because all of them are important, but this is the one where you do the actual optimization and standardization of machine learning algorithms on microbiome data from the benchmark test here. And all these feed into the dissemination and training working group, where we have training schools and we also did conferences and so on. So we met quite a few times before COVID, and now it's all Zoom meetings, but we hope to get back to physical meetings too. A very nice part of this, of this cost action, are the so-called short-term specific missions. So this is where PhD students and postdocs can go between member labs and spend a few weeks paid by the cost action in other collaborators' labs and they work on these topics. So some of the outputs from this, because we haven't really finished the optimization, that comes quite late. We published recently 2 papers in Frontiers of Microbiology on the current state of the art. You can look into those there. In a separate review I was a part of, we published, we described how machine learning can be used for precision medicine in cancer, which is another very important subject. And in terms of outputs, midway through this cost action, we will soon have a dream challenge, which is where people outside the cost action machine learning experts can compete with their own favorite approaches on a kind of a blind data set, and then we see which one gets closest to the answer. We also have a couple of bioconductor R packages here, and I'm also a member of a Horizon 2020 action consortia, which is separate to this cost action, which also aims to standardize the field of microbiome. So, for instance, define what is a healthy microbiome, and also on a more methodological basis. So last, I just want to plug a company, spin-out company I'm involved with. It's called SeqBiome, where we do sequencing and microbiome analysis for both academia and industry. So it's a spin-out about 2 years ago from HC Microbiome Ireland. So me and colleague CTO Paul Cotter, we have it's based on our microbiome experience through the years, and we have state-of-the-art sequencing and analysis pipelines, and we already have quite a few blue chip clients in food and pharma. So just to finish, just to thank a large number of people without whom it would be impossible to have done these studies. This is my lab. And also some previous lab members and lots of funders as well from Canada, Ireland, and UK. So with that, I'm happy to take any questions.
So thank you very much, Marcus. It's quite impressive. You covered quite a lot also. Are there questions? I know we have a limited number of minutes for questions, but there is still some. So if you just use the opportunity while you have it. Just go ahead.
Hi, Marcus. John Mark from BioMe. You mentioned that 2 time point was better at predicting than a single time point. And I was curious as of how you use these 2 time point. Did you aggregate them? And if yes, how did you do that? Or did you consider them independently in the model?
Yeah, that's a good question. What we did is we had an OTU table, and we had 3 time points for each person, and we looked into consecutive time points within each person. That sounds bad. That sounds strange, but that's so. We basically took an OTU for each OTU. We took the ratio of the abundance between time point 1 and time point 2 from the same person, and then time point 2 and time point 3 as well. So we use this ratio rather than the relative abundance from one time point, and we use that as an input to the XGBoost algorithm.
And a follow-up question: why do you think this ratio is so much better at predicting rather than a single time point?
If you recall from one of the previous slides on the same study, we had much more variation, microbiota variation between transitioning patients, those that went between relapse and remission within the 6 months. And we think that information is important for the algorithm as well, to kind of larger variation, and then to predict that compared to those who didn't move between different states of disease.
I see. Thank you very much.
Thank you.
So, thank you. Just the microphone is open. If not, I can continue. You both showed that there seemed to be like a relationship of those patients with less potential for butyrate production. They also ate less fiber, while overall there was still a limited association between the overall diet and the diversity and so on. What do you think? Is the low butyrate phenomenon in microbiota, do you think is diet driven, or is it just secondary to disease, or what do you think?
I think with complex diseases like this, it's hard to give a straight answer. I think the fact that you have IBD promotes certain, and you have more inflammation in your gut promotes certain bacteria, which are less known to produce butyrate. And you might also have, on the other hand, so for instance, you have people with Crohn's disease who, if they're quite active disease, they're recommended not to eat very fibrous food because it's can be constrictions in the gut and so on. So that means they will also have a low fiber diet, and they will promote less bacteria that can turn these dietary fibers into butyrate. So it's a bit chicken and egg, but it can also be both at the same time.
Very good, thank you. It's a complex question. Another question I had is that we're all interested in, well, with the opportunities within machine learning, artificial intelligence are of course great, but I guess the majority of us will be looking at studies of, say, 100 or 200 maximum. And then to what degree do you think those types of statistical techniques will be relevant or important to understand those results?
Sorry, are you asking whether it's?
Do you think if you have like 100 samples or 150, 200 samples, should we spend a lot of energy into more advanced statistics? I guess that's my question.
Yeah, as you mean, it's always that challenge with the number of samples, and with sequencing technologies becoming cheaper, it is possible to have more. But in some cases, like we have a study like the one with collect biopsies, even if it was very cheap, it wouldn't be possible to, from a single hospital, get all these biopsies. So it is a challenge. My group will be looking into ways of boosting the sample numbers synthetically, in a way, by simulating healthy microbiome samples. There are various machine learning methods you can do that, which could be one way of improving classification accuracy and so on. But that's quite we don't know if that will work yet.
No, very good. Still no more in the audience. Mikael?
Hello, my name is Mikael from Oslo University Hospital. Thank you for a great talk. I might have missed it, but in regards to your first study, did you grade the colonic biopsies by severity, by endoscopy or histology, and look at taxonomic association or diversity association?
Yeah, that's actually a question we got from the reviewer back in the time. So that wasn't done. We had kind of a very experienced gastroenterologist, Professor Fergus Shanahan, and it was more visual inspection of the inflamed. We weren't looking into grades of inflammation. It was either or. Apparently, when you're in there, it's very clear whether a region is inflamed or not. And he only picked those biopsies from regions that were very clear cut, either inflamed and non-inflamed. So it was binary in our case.
Thank you.
Okay, final question then from Maria.
Hi. I'm wondering about how important do you think the resolution is to distinguish between UC and CD?
Maria, do you mean the resolution of taxonomic level?
Yeah.
Yeah. I think it's important. If I go back to this earlier plot here, where here you can see, if you look at the biplot here of B, so B is short for Bacteroides. You have Bacteroides dorei up, Bacteroides vulgatus down. And and this is based on the 16S sequence of V3, V4 region, which in many cases is only able to go down to genus level. But we have developed a tool called Spingo, which does species classification based on 16S sequences, but it only does it when the variation within a certain genus allows that. And in this case, I think if we only used genus, we wouldn't detect these differences between Vulgatus and Doreae because both are from the same genus. So that's one example where species classification is important. And I know there's also important subspecies classification that we couldn't do here because it's all 16S, but the Colibacterium prosnitsia and Prevotella copperi, they all have important subspecies classification as well with very different differences in function. So ideally you should do this with shotgun, but we tried to get shotgun data from these biopsies, but there simply wasn't enough bacterial DNA to do it.
Okay. Thank you very much, Marcus. I think that was actually a very interesting observation in the end, and thank you so much for a great talk. And I know you are busy very soon, so thank you for joining us, and best of luck.
Thank you. It's a pleasure.